Hi class, and welcome to our final lecture on the urinary system. So we'll end this semester talking about the urinary system, chapter 24. You should know the function of your kidneys, and this is a review from anatomy. So the reason why these slides are kind of long is I include the anatomy in here, but I will pass over that. But your kidneys help to regulate your total water volume, the total solute concentration. They regulate ion concentrations in fluid that are extracellularly or outside the cells. They ensure long-term acid-base balance of the body. They excrete metabolic wastes, toxins, drugs, and they produce two hormones, erythropoietin, which stimulates red blood cell production to help regulate blood pressure, and renin that helps to, and actually these are switched, so sorry about this. Renin is actually the one that regulates blood pressure, and erythropoietin is the one that regulates red blood cell production. So those two should be switched. They also activate vitamin D and carry out gluconeogenesis, so producing glucose if needed. And then here are the parts of the urinary system that you should know well. A picture like this, you should know, you have two kidneys, the ureters leading to the bladder, the urethra exits the urine out of the body. So you should know the gross anatomy of the kidneys well, which is a review from anatomy. So I'm going to pass over this that goes through here. Specific parts of anatomy you might not need to know, but you should know generally, what the renal cortex is, the renal medulla, the pyramids, the papilla, the lobes, the funnel-shaped tube that connects with the ureters, the renal pelvis, and the minor calyxes drain from each of the pyramidal papilla. Minor calyxes join with major calyxes. You should know this flow of urine through the kidney well, and let's see if we can see a picture. So reviewing basic anatomy of the kidneys will be important as well. Here's a look at some inflammations of the renal pelvis or the entire kidney. I don't think I asked you questions about this. You should know blood flow through the kidney, so how the renal arteries deliver blood to the kidneys each minute, about 1,200 milliliters, which is a lot, and then the arterial flow through the kidney as well. So here's a good review of the blood vessels of the kidney and how they lead the blood through the kidney, which is important because your kidneys are the main filtering mechanisms of the blood for the body. The nephron is the structural and functional unit that forms urine, and you have about 1 million nephrons per kidney. There's two parts to the nephron, the renal corpuscle and the renal tubule. And again, you should review this really well because then we'll get into the physiology behind urine formation in a little bit. So the glomerulus is shown here, the glomerular capsule. Here are the layers of the glomerular capsule. The visceral layer, so the glomerular capsule has podocytes surrounding it, which kind of makes a membrane for filtration to occur. You should know well the renal tubule and these three parts, the proximal convoluted tubule, the nephron loop, and the distal convoluted tubule that will drain into the collecting duct. So please review renal tubule, collecting duct, proximal convoluted tubule. This is kind of the powerhouse of the kidney. This is the main reabsorption center. It'll be mainly in the cortex of the cell. It will have cuboidal cells with dense microvilli that form a brush border to increase surface area. So you should know the structure of the cells that make up each of these tubules as well. So your proximal convoluted tubule is made up of these cuboid shaped cells with the brush border. And you might see a picture of these cells on your lecture exam. So you should know that these cuboid shaped cells with the microvilli are part of the proximal convoluted tubule. You should know that the parietal layer and then this visceral layer will make up the glomerular capsule. The nephron loop has a descending limb and an ascending limb. There's thick and thin parts to each descending and ascending limb, depending on what they secrete or absorb at the various parts. And they will be very more thin segment cells of the nephron loop. So again, kind of looking at these pictures and being able to match pictures to where in the nephron they're located. Then the distal convoluted tubule has cuboidal cells, but with very few microvilli, and they function more in secretion, so secreting things back into the urine to make it more concentrated. And this is a look at the distal convoluted tubule cells. They are cuboid in shape, but they will not have as much microvilli to them. All right, then the collecting duct, you have principal cells and intercalated cells. The principal cells maintain water and sodium balance, and then the intercalated cells help to maintain acid-base balance of the blood. Your collecting ducts will receive filtrate from all the nephrons, run through the medullary pyramids, and then fuse together through the papilla to drain or funnel that urine that's formed into the minor calyxes. So here are the collecting ducts, which are made of principal cells and then intercalated cells. So if you're looking at a picture for collecting duct, you'll see two types of cells that are next to each other. So here's a look at the location and structure of the nephrons. And again, you have a picture on your lecture exam. You have a question on your lecture exam matching parts of the nephron to specific cell structure that makes up the layer of the cells that form the tubule. And I want you guys to know that because it ties in with the physiology, because it'll help you remember and understand what substances can pass through these cells then, because at different parts of the nephron, different things will be secreted into the urine to create it more concentrated to the filtrate, and different substances might be absorbed out of the urine back into the bloodstream, depending on where along this kind of scheme is happening. So you can have cortical nephrons or juxtamedullary nephrons. Here's a look at on the left is a cortical nephron, and then the juxtamedullary nephron. The renal tubules are associated with two capillary beds, the glomerulus and the peritubular capillaries. So the glomerulus will be where filtration occurs at first. The peritubular capillaries surround the distal and convoluted tubules. The vasa recta specifically surrounds the nephron loop, and that would be a part of a juxtamedullary nephron. So here is the glomerulus, the first of the capillary beds. This capillary is specialized for filtration. The afferent arteriole will bring blood into the glomerulus, and the efferent arteriole leaves the glomerulus. Blood pressure in the glomerulus is always extremely high because the afferent arteriole has a much larger diameter, and that high pressure ensures filtration of the blood to occur in this glomerulus. The peritubular capillaries will cling to adjacent renal tubules. They will empty into venules, and the peritubular capillaries will surround the proximal and distal convoluted tubules, and they're adapted for absorption of water and solutes from the filtrate back into the blood. And then the vasa recta will run parallel to the long nephron loops, and they will function in the formation of concentrating the urine. So here's a look at peritubular capillary beds surrounding the distal convoluted tubules. So here again is a look at the nephron, and you should know well the flow of filtrate through the nephron, starting from filtration occurring in the glomerulus, traveling through the proximal convoluted tubule, down and up the loop of Henle, and then into the distal convoluted tubule before the collecting duct takes all that urine formation. So a little bit of review from anatomy before we kind of get into the physiology of urine formation. Each nephron has what we call a juxtaglomerular complex. It involves modified portions of the distal portion of your ascending limb of the nephron loop and the afferent arteriole, and this JGC is important in regulating the rate of filtrate formation and blood pressure. There's 3 cell populations in this juxtaglomerular complex. We have the macula densa cells, which contain chemoreceptors that sense the sodium, the salt content, basically, of the filtrate. You have granular cells, or JG cells. These are enlarged, smooth muscle cells. They will act as a mechanoreceptor to sense blood pressure, and they will contain secretory granules that contain the enzyme renin, which will help regulate blood pressure. And then extraglomerular mesangial cells may just pass signals between the macula densa and the granular cells. So here's a look at the juxtaglomerular complex as shown here, and the 3 types of cells. Again, as we look, the big one I want you to know are the granular cells, which are shown here, and they will secrete renin, which will help to regulate blood volume. We have extraglomerular cells and then the macula densa cells. Here's the afferent arteriole bringing blood into the glomerulus, and this glomerulus is again a kind of a series of tightly wound capillaries where filtration will occur. So what that means is blood gets kind of pushed in here at a very high pressure, and then filtrate gets pushed out through the filtration membrane, and that filtrate will travel into the proximal convoluted tubule. From here, it goes through the tubule system, from the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and that in the filtrate, different substances will be absorbed back into the blood or secreted into the filtrate to create the concentrated urine. So about 180 liters of fluid is processed daily in the kidney, but only about 1.5 liters a day of urine is formed. The kidneys filter your body's entire plasma about 60 times a day, and they consume about 20% of your oxygen at rest. The filtrate, which is produced by glomerular filtration, is basically the blood plasma minus protein. So proteins and red blood cells should never be in your urine because they're too large to pass through that filtration membrane. Urine then is produced from the filtrate, and it's less than 1% of the original filtrate. So most of your filtrate, water, different substances, will actually get reabsorbed back into the bloodstream, and about 1% of the original filtrate will actually go on to form urine, which will be excreted from the body. And your urine will contain metabolic wastes and other unneeded substances that your body doesn't need. So now we get into the physiology of urine formation, and we divide it into 3 kind of steps: glomerular filtration, which produces cell and protein-free filtrate. That's important to know because whatever gets through that glomerulus and gets filtered should never be proteins and should never be red blood cells. So if you are finding blood in your urine, or it can get tested for proteins, there's usually a problem with that glomerulus, the glomerular filtration step. Tubular reabsorption. So about 99% of substances from the filtrate get returned back to the blood. And then tubular secretion selectively moves some substances from the blood to the filtrate in the renal tubules and the collecting ducts to create that concentrated urine. So these are the 3 main simple processes of urine formation. Glomerular filtration occurs at the glomerulus, which creates the filtrate. Then that filtrate travels through the tubule system, proximal convoluted tubule, loop of Henle, distal convoluted tubule, into the collecting duct, whereas most of the filtrate will get reabsorbed. That's called tubular reabsorption, back into the peritubular capillaries, but some substances will get secreted back into the urine to make it more concentrated. Glomerular filtration is a passive process. No energy is required. That's because we have lots of pressure called hydrostatic pressure and other different pressures that will force fluids and solutes through the filtration membrane into the glomerular capsule. The filtration membrane itself, it is made up of these 3 layers, and I'm just going to go ahead and show you a picture of this. So here is blood coming into the glomerulus is at extremely high pressures. That high pressure forces the filtrate out of the blood and into the capsular space where it's caught as filtrate and then travels through a tubular system. Here's the filtration membrane. Let's show, we have kind of slits in those capillaries. We have podocytes with little feet and slits that allow substances to pass through. So this is what makes up the filtration membrane. Again, only small substances can get through as filtrate. Proteins are too big to get through, as well as red blood cells. So again, if you're finding blood or protein in your urine, there's a problem at this filtration membrane in the glomerulus. So here's a look at the filtration membrane, the 3 layers and the blood and then the filtrate. And again, only small substances can get through these slits to get into that capsular space. Macromolecules can get stuck in the filtration membrane and they'll be engulfed or eaten by some of your mesangial cells. The filtration membrane does allow water, glucose, amino acids, and nitrogenous waste to get through. Plasma proteins will remain in the blood to maintain pressure to prevent loss of water in the capsular space. And again, if you find proteins in the filtrate, that indicates a membrane problem. And then we go through pressures that affect filtration, and you don't need to know these. What I just want you, I'm just going to show you one picture. Basically, there's pressures coming out from the blood, and then there's pressures forcing against the blood back into the glomerulus. But basically, net pressure is about 10 millimeters of mercury. When we add all these pressures together, forces pushing filtrate out, forces pushing back on the filtrate coming out, we still have a net outward pressure that's the greatest pressure of about 10 millimeters of mercury. That's forcing this filtrate out of the blood into the capsular space. And again, that's a passive process. The glomerular filtration rate is just the volume of filtrate, formed per minute by both kidneys. It's about 120 to 125 milliliters per minute. It's proportional to the net filtration pressure, the total surface area available for filtration, and the filtration membrane permeability. And then we have different intrinsic controls that maintain that glomerular filtration rate. It will affect blood pressure, and there's also extrinsic controls to help maintain systemic blood pressure. And I don't ask you too many questions about the renal autoregulation of this glomerular filtration rate, but it's under renal control as well as other control as well. The sympathetic nervous system helps to control under extremely low extracellular fluid volume. So if there's low blood pressure, norepinephrine will be released to cause vasoconstriction, constriction of afferent arterioles, basically to try to increase blood volume and blood pressure. The renin-angiotensin-aldosterone mechanism is the main mechanism in the body for increasing blood pressure. If you go to the hormone, I think the hormone talk, you'll learn a little bit about the renin-angiotensin-aldosterone mechanism. It has a direct stimulation of the granular cells by the sympathetic nervous system, stimulation activated by the macula densa cells, and there's reduced stretch of the granular cells. So any sort of decrease in systemic blood pressure will kind of put this system into gear to try to increase systemic blood pressure again. Other factors affecting glomerular filtration rate are shown here, which I don't ask you main questions about. And then this is a summary of regulation of this glomerular filtration rate. Anuria is abnormally low urinary output. It may indicate that glomerular blood pressure is too low. Renal failure can also be from situations when you have maybe inflammation of the nephrons, a transfusion reaction, or some sort of crush injury or crash injury. So then step 2 in formation of urine is tubular reabsorption. It quickly reclaims most of the tubular contents and returns them to the blood. Almost all organic nutrients are reabsorbed, and water and ion reabsorption is hormonally regulated and adjusted. So we have active and passive tubular reabsorption. And again, tubular reabsorption is reabsorbing substances from the filtrate back into the bloodstream. We have different routes for tubular reabsorption, whether they go transcellularly directly across the cells or paracellularly between the cells. You don't need to know that, but just know that there's different routes for reabsorption depending if we're absorbing water and solutes. They can go through the cell or between the cells. Sodium transport. Sodium is the most abundant cation in filtrate, and transport of sodium requires energy. So we have a sodium-potassium pump, and that's what this secondary active transport is allowing for. You should know in general, the proximal convoluted tubule is the site of most reabsorption. All nutrients, glucose, amino acids will be reabsorbed back into the bloodstream. About 65% of sodium in water is reabsorbed, many of your ions, almost all of the uric acid, and about half of the urea is reabsorbed back into the proximal convoluted tubule, but will be later secreted back into the filtrate. You should know what happens in the nephron loop. In the descending limb, water can leave, but solutes cannot. And in the ascending limb, water cannot leave the nephron loop, but solutes can. The thin segment is passive to sodium movement. The thin segment has different symporters. So you should know basically what happens in the nephron loop as well. The distal convoluted tubule and the collecting duct are under hormonal control. So antidiuretic hormone that's released from your posterior pituitary gland, it basically causes cells to insert what we call aquaporins, which help to increase water reabsorption. So if you have an increased level of ADH, this will cause an increased water reabsorption. So you're retaining more water. You're making the urine more concentrated, and because you're retaining more water, you're raising your levels of blood volume and blood pressure in the blood. Aldosterone targets the collecting ducts and the distal convoluted tubule. It promotes the synthesis of apical sodium and potassium channels. So as a result of aldosterone, little sodium leaves the body. So basically you're retaining sodium, so you're also increasing blood pressure levels by retaining sodium and decreasing potassium levels. Atrial natriuretic peptide is released from the heart. It will reduce blood sodium, resulting in decreased blood volume and decreased blood pressure, and it's released by cardiac cells if blood volume or blood pressure is too high. And parathyroid hormone also acts on your distal convoluted tubule to increase calcium reabsorption. So here's kind of a basic summary of tubular reabsorption and secretion throughout the various parts of the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. These next two slides I include because it's probably a great review summary of what happens in the proximal convoluted tubule, the nephron loop, the distal convoluted tubule, and the collecting duct. So studying these two slides will help you with several questions on the lecture exam. Tubular secretion then is step 3. This is reabsorption in reverse, meaning we're secreting things back into the filtrate. It occurs almost completely in the proximal convoluted tubule when selected substances are moved from the peritubular capillaries through the tubular cells back into the filtrate. So this is secretion, moving things back into the filtrate. This is important for disposing substances, drugs, metabolites that are bound to plasma proteins, eliminating undesirable substances like urea, uric acid, ridding the body of excess potassium ions, and controlling blood pH by altering the amounts of hydrogen or bicarbonate ions in the urine. Diuretics, so chemicals that enhance urinary output. So a diuretic makes you urinate more frequently. Anything that inhibits ADH, because remember, ADH retains water. So anything that inhibits ADH, like alcohol, will be a diuretic. Sodium reabsorption inhibitors, caffeine, and drugs are diuretics. Osmotic diuretics are substances not reabsorbed, so water remains in the urine. So for example, in a diabetic patient, if you have a high glucose concentration, this pulls water from the body and causes you to urinate more frequently as well. A urinalysis is when we examine urine for the sign of disease. This is really common. It can also be used as a drug test, testing for an illegal substance. Assessing renal function is really important, and it requires blood and urine examination. Renal function can be assessed by measuring nitrogenous waste in blood, but to determine renal clearance, both blood and urine are required. And that's a quick summary of the urinary system. So again, look for a lot of matching questions on your lecture exam, looking at the different cell types that make up the various parts of the nephron, and then knowing the basic functions of the nephron itself will be important. So thanks for listening, guys. Thanks for a great semester, and I'm sure we will see you again soon.